We will continue with the secondary structure of peptides. So these can be either alpha helix, beta pleated sheet, turn, or loops. These are some unordered and coils as well. These are some unordered parts of the peptide sequences. Typically, the turns are shorter, maybe 3 amino acids sequences, and coils are longer, unorganized or disordered regions of proteins. So, as for the particular types of secondary structure, the alpha helix is the most common, and it's organized form of helix where and it is enabled due to hydrogen bonding between the particular floors of the helix. The side chains of the amino acids are pulled out of the alpha helix. The other most important type of secondary structure is the beta pleated sheet. Where the amino acid sequence is linear, the side chains go either up it means above or below the beta pleated sheet, and in the beta pleated sheet there are actually two amino acid sequences connected via the hydrogen bonds, either in parallel arrangement or anti-parallel arrangement. So in the picture here there is the anti-parallel arrangement. It means we have the nitrogen end here and C end here, whereas in the other side there is the C end here and nitrogen end of this sequence here. So this is so-called anti-parallel arrangement in beta pleated sheet. Okay, I don't like to focus much on the protein folding, which is a complicated process. But I would like to show some super secondary structures, so-called structural motifs, for alpha helix or beta pleated sheet. So, for example, the helix turn helix involves one alpha helix with short loop. This is the disordered region of the amino acid sequence, and then the chain continues with another helix to the nitrogen end. I already mentioned turned as shorter regions, whereas loops or coils are longer disordered regions. In case of beta hairpin, there is the typical anti-parallel arrangement. In this case, one linear chain going to turn and then continuing down again, forming a sort of hairpin. And in case of the Greek key, there are actually 4 parts of the beta pleated sheet with the turns and coils. So you can see it in the protein structure, this structural motif. So here is an example of a complex of protein with DNA, double helix. And you can observe in the structure of the protein depicted in green the helix, loops, and helix continuing in the protein sequence. Apart from the structural motifs, we have or we can recognize in protein structure so-called structural domains, which are the structural units of tertiary structure. And in the protein, they can be typically only alpha domains, which means only part based on the helices, or only beta domains, parts based on beta pleated sheets, or combinations of alpha and beta domains that can be either alternating or can be separated in the regions. So we have one example of a human beta-2 adrenergic receptor that is created only from alpha helices. So it means alpha domains. Here are some other examples, for example, cytochrome B or human growth hormone, that is peptide. And as for the domains containing only beta pleated sheets, we have an example of up and down sheets creating a sort of barrel. It's term used in protein structure. As for the combination of alpha and beta domains in one protein together, so we have an example of specific so-called thin barrel where the beta pleated sheets are organized in the structure in turn with alpha helices. And it can create different types, for example, the Rossmann fold or alpha beta horseshoe fold. So it depends on the overall form of the protein or organization of the protein. And the last type of structure is the ternary structure, and it means that two polypeptides subunits can be associated in one peptide via van der Waals interaction or any other non-covalent interactions. And maybe we have not mentioned the disulfide bond, which can happen due to presence of cysteine in the proteins. But the quaternary structure is based mainly on the van der Waals interactions. So We would like also to mention a little bit something about enzymes, about the active site of enzymes. And it can have different role. The amino acids in the binding site can have different role, either binding or catalytic. So I would like to show some examples. I have mentioned already a little bit the induced fit mode of binding substrate into the enzyme. It means when drug is binding or any substrate to the binding site, the enzymes undergoes conformational changes. So it fits better around the substrate or drug to enable better the interactions and to get the amino acid residues responsible for the binding closer to the part of the ligand or substrate. So we don't talk anymore about the lock and key hypothesis, but we talk about the induced fit mode of binding between the protein and drug or substrate. There are specific functions for some amino acids. So we have already mentioned histidine as a relatively weak base. So it can be at physiological pH ionized or non-ionized, which is depicted in the picture below on the left side. Glutamic acid and tyrosine can act as a proton source. Aspartic acid and asparagine can act as proton donors or acceptors. As for the lipophilic residues, for example, in the sulfanyl group in cysteine or hydroxy group in serine, there is the lone electron pair, and these functional group are also willing to form a covalent bond. So typically, the sulfanyl group in cysteine is creating the structure due to the structure of protein due to the disulfide bonding, and the hydroxyl of serine, for example, can form covalent bond with drugs, covalently bond drugs. And one remark to lysine: when it is in hydrophobic pocket, it can be also non-protonated, although it is a positively charged amino acid. So one example of catalysis. So this is an example of hydrolysis of peptide both, for example, in diet protein. So we have so-called catalytic triade, which is a triade of the three amino acids in the binding site. So it's chymotrypsin. And you can see that serine plays the role in binding to the carbonyl carbon under the stabilization of histidine that can accept the proton from serine, so that serine is able to create the covalent bond with the carbon in the protein. The protonized histidine then affords the proton to the amino part of the peptide under release of amine, and water molecule is approaching to the acyl bound to the serine. Histidine is assisting again with accepting the proton from water, and the carboxylic acid is then formed and released from the binding site. You can see that aspartic acid creates a complex with the histidine, playing role in the stabilization of the histidine in the binding site. So every amino acid has some specific role during this catalytic process, which was example of peptide bond hydrolysis. There might be also some other components in the active sites of enzymes, so-called cofactors. These can be, for example, metal ions or any coenzymes. As for the metal ions, they are present in metal enzymes, and they play a role in coordination of the substrate in the catalytic in the binding site. Iron is, for example, involved in hemoglobin. There might be also manganese, copper, or zinc. You may remember zinc is involved in angiotensin converting enzyme, or any matrix metalloproteinases, for example. And apart from metals, there might be also coenzymes. They are derived from water-soluble vitamins, and they play a role in redox reactions, for example, NADH or NADPH, or FAD, FMN. I have one example of protein that involves the metal in the binding site, and it's the iron in the binding site of nicotinamidase or pyrazine amidase, and it plays the role in hydrolysis of pyrazine amide, which is this molecule known as anti-TB drugs, anti-tuberculosis drugs, that needs to be hydrolyzed after entering mycobacteria. So the activity of enzymes is regulated by internal control. It means inside the cell. It can be controlled by level of the resulting products from the catalysis. It can be regulated by some chemical messengers that enter the cell and it can be regulated also by some other molecules that are not able to bind in the active site, but they bind, they differ in the structure, and they bind to the allosteric binding site, and they cause the change in the conformation of the binding site, which can then disable the function of the enzyme. We can talk also about isoenzymes. These are enzymes that have similar function, but they can differ in substrate specificity or tissue distribution. One example can be 11-beta hydroxysteroid dehydrogenase. You may remember this enzyme from pharmaceutical chemistry, where it plays the role in reduction or oxidation of cortisol and cortisol respectively. So this enzyme is tissue specific. There is one isoenzyme present in liver and another one in kidney. So let's talk now about the DNA structure, which I suppose will be also a revision for you. So we have the primary structure in DNA. That is consisted of deoxyadenosine, deoxyguanosine, deoxythymidine, deoxycytidine connected through the phosphate groups. And the secondary structure is enabled due to the hydrogen bonding that are formed during pairing of the bases, creating the double helix of DNA. And there is also some tertiary structure, which is coiling of the secondary structure catalyzed by topoisomerases. These are enzymes that can be also target of drugs. For example, some anti-cancer or antibacterial agents. So let's have a look on the picture. So from the left side, there is the structural, the precise structure, then simplified structure of the deoxyriboses bound to phosphates, where the deoxyribose carries the base. And on the right side, you can see the pairing via the hydrogen bonding between thymine and adenine and cytosine and guanine. There are either 2 or 3 hydrogen bonds between them. And you can see here the double helix, showing also the major and minor grooves in the structure of the double helix. So you know the function of DNA. It's it's the source of genetic information that can be it can be replicated, and always 3 nucleoside codes for amino acids. So the replication, the genetic information is copied from DNA to RNA. The structure of RNA can be primary and secondary. The primary is very similar to DNA. However, the difference is that there is not deoxyribose, but ribose involved in the structure, and uracil is the replacement for thymine in the structure. The secondary structure can look different for different types of RNA, and we have three types, which is the messenger RNA, ribosome RNA, and the RNA. So you can see the process of replication. The messenger RNA goes to a ribosome where the ribosomal RNA is present, and the RNA carries the particular amino acids, and it is complementary added in the process called translation. You can see the structure of the RNA, transfer RNA here, and the coding of particular amino acids here in the circle by combination of the 3 bases in the structure. So the nucleic acids can be target of some drugs. So I have already mentioned some alkylating agents, but there are also intercalating drugs creating non-covalent bond with DNA. There are the topoisomerase inhibitors, or so-called poisons. There are chain cutters that stop the synthesis of nucleic acids, and so-called chain terminators. We have some drugs that are used interacting with RNA, either interacting or binding to ribosomes, or so-called antisense therapy, which is the field of biological drugs. And at the end, I have listed some methods or assays that are used for receptor ligand interactions, checking or exploring. They can be generally, they can be based on labeled assays or label-free assays. One example is the surface plasmon resonance. So I have highlighted some methods in bold here that you can hear of them. The other 2 methods belong to thermodynamic binding assays, and it's the thermal denaturation assay and so-called isothermal titration calorimetry. Apart from these 3 methods, we have already mentioned the structure-based ligand binding assays in the last lecture, and maybe you will hear them about them in some of the following lectures. and these are nuclear magnetic resonance and X-ray crystallography. For these two methods, we can directly observe the interaction of the ligand with the target. So these two methods are structure-based ligand binding assays, and we can see the details of the interactions, which is not the case of the other methods. And I have listed the three methods highlighted in bold here. Surface plasmon resonance, that is based on measurement of refractivity or reflectivity. And it is based on a surface where the proteins are bound via some specific bonds. So we have the proteins, and in case the ligand are bound, there can be observed a difference in the refractivity or reflectivity. So we can see the change under binding the ligand to the protein. I do not like to go into the detail of this method. All the terms that are in blue, there is an interactive link to Wikipedia. So if you want to be informed more, you can go to English Wikipedia. However, I subtracted this from some scientific publications. Isothermal titration calorimetry is based on measurement of heat. So we measure the calories during the binding of the ligand to the target, to the protein. So it's the isothermal titration calorimetry. And the last method is thermal shift assay. And in this assay, we measure the protein unfolding temperature. So the unfolding temperature would be different under a condition when the ligand is bound to the protein in comparison to the state it is not bound. So
there is no interaction with the
folding of the protein.
There is no bond, yeah?
Actually, during protein synthesis, the proteins are somehow folded together into the final shape, and using the temperature, you can measure the temperature of protein unfolding. I do not perform this method by my hand, so
that's why I have seen it in the articles.
You can go either to Wikipedia via the interactive link, or to this thesis that is available in the internet, freely available. So if you want to read more details, we just want from you, sorry, your homework. You can run a video from the PDB website. It's an educative website. When I go back to the methods for measuring the interaction, so we definitely would like from you to know the structure-based ligand binding assays, which are also methods for 3D structure determination of proteins, for proteins. And in publications, these three methods highlighted in bold are also mentioned, so you may know some principles, but I think these three methods are not involved in the test, but I should not tell you.